So we're very, very happy to listen today to the voice of industry. We thought it was really important to listen to why industry needs biomarkers as we do, and to listen to their point of view. And we're happy today to welcome Janet Nicholson, who is pharmacologist. She has studied in UK. I think you have done your PhD in Cambridge and your postdoc in the States at the University of Michigan. You've been working in UK at Pfizer before joining in Germany BI, where you have been the director of research in precision psychiatry, which is very close to our focus of interest. And you're now working at the Department of Translational Medicine in BI. So welcome. Thank you very much to have accepted to give this conference in front of the network of expert centers and the participants to our program project in precision psychiatry. And the floor is yours.
Absolutely. Thank you. Thank you, Mariam, for that nice introduction. Yes, indeed. So good afternoon, everyone. It's a pleasure to be here. I'm here today to tell you about some of the work that we do at Boehringer Ingelheim in the field of precision psychiatry. And biomarkers are absolutely fundamental to everything that we do in this field. And so what I would like to share with you this afternoon is some of the work that we are doing, and to describe to you why biomarkers are so important, how we use them, and what sort of examples we have of biomarkers that we are using to advance our pipeline portfolio for precision psychiatry. So I will start just with a very brief introduction here. We are all aware of the medical need in psychiatry and how tremendous that is. It's estimated that around 800 million people in the world are living with a mental health disorder. Increasingly young people, including adolescents, are being affected. And it's a serious, serious crisis. It has widespread consequences, not only for the individuals themselves, but also for families and loved ones. And when we look at the impact on society, more broadly, the economic burden of mental health disorders is also considerable. It's estimated to exceed 4% of gross domestic product across OECD countries, with direct and indirect costs worldwide estimated to around $2.5 trillion US. Absolutely incredible. What you see on the lower part of this slide is an overview of the pharmacotherapy of mental illnesses over the last 70 plus years, dominated by serendipitous findings with still incomplete understanding of modes of action of these drugs, which are being prescribed, as you very well know, based on diagnoses using clinical diagnostic systems such as DSM and ICD-10, 11, that have been designed to provide a common language, very important common language, which is based on observable signs and symptoms. However, it's important to note that these classification systems were not intended for research purposes. We are now, fortunately, at a stage where there have been, in the field, significant technological advances that are enabling an increased understanding of the underlying neurobiology of mental disorders. For example, advances in neuroimaging. We also have precision technologies such as optogenetics that can be used in the preclinical setting. We've got advances in human genetics, behavioral measures that can also be used as biomarkers. And in addition to such technological advances, there has also been a more formal recognition of the need to be more systematic and rational in research and drug discovery activities since the publication of the research domain criteria, which came from the National Institute of Mental Health over a decade ago. And it's really this RDoC combined with the technological advances in neuroscience that have driven a shift to recognize psychiatric symptoms as manifestations of malfunctioning brain circuits. It's more than a decade ago, as said. Significant progress has been made in that time, although there is really still a lot to do. What you can see here is it's really illustrating why it is the case that the classical diagnosis systems do not support research and development. So heterogeneity of those disorders is one major challenge for drug discovery and research. And if you look here at this table, you can see a list of symptoms, and those where there is an asterisk attached, this asterisk denotes the symptoms that are part of the diagnostic criteria for MDD. So you can see that there are quite a number of symptoms that are associated with MDD, and not only a number of symptoms, but also they are very, very heterogeneous. When you look at insomnia or difficulty concentrating or hypersomnia, psychomotor agitation and so on, depressed mood, a lot of symptoms, but also very, very diverse symptoms which surely have a very different underlying neurobiology. So heterogeneity is there, which doesn't help us. And in addition to that, we have a lack of specificity. So here on the right-hand side of this table, you see the number of diagnoses that are associated with each of these symptoms. Insomnia, 22 different diagnoses where insomnia plays a role. Concentration, 17, and so on. So you see it's heterogeneity and lack of specificity of these disorders that really are a stumbling block for us in research. So what we would like to do, our approach to this, is to break down the DSM categories into smaller, measurable components, circuits and systems that we can really interrogate and use then to improve the feasibility for research and development. So this is where we are now. We are on hopefully a path to a more precise psychiatry. We need DSM-5, and we will, of course, we won't move away from DSM-5, but that combined with then the RDoC-inspired approach to break those disorders down into more measurable clusters of patients who share perhaps a common symptom and a common underlying neurobiology, and use that then to back translate into the preclinical setting, and then we can use that then to forward translate more specific approaches that then can be used as part of a precision psychiatry approach, which is aiming in the future, this is our hope for the future, to be much more rational, to rationally deliver then the right drug to the right patient, and really at the right time point for that specific individual. So that's what we are aiming to do. And just in terms of also just a little bit of background to what we are doing at Boehringer Ingelheim, we are aiming to address core symptom domains that we have selected. And we have selected particular symptom domains based on scientific rationale. We are balancing biological understanding with clinical need. These, we feel, are areas that are feasible for us to tackle based on the knowledge that is already there in the clinical and preclinical literature, where it's possible to target cognition, for example, working memory, episodic memory, cognitive flexibility, and executive function, based on an understanding of certain brain regions. Prefrontal cortex, cortical EI imbalance, for example, is a circuitry that can be targeted. Similar examples here also for emotion, for impulsivity, which I will focus on in today's presentation as an example. Also motivation and reward. These are all areas where there's some understanding of specific circuitries that are involved that lead to then these symptom domains. And these are the circuitries that we can really explore then very precisely in the preclinical setting to identify then targets that we can then use as the starting point for new therapeutic concepts. So this is an overview of how we work in drug discovery. The starting point is preclinical research, where we identify new therapeutic concepts and new molecular entities using some of the precision tools that I've already mentioned. And this is then, these new concepts and molecules are then handed over to the department and clinical pharmacology, where we perform then the first in human studies, where we want to demonstrate that these new drugs are able to get into the brain, that they are able to engage with the drug target, and that they are able to exert efficacy. In our very early clinical trials, these are small studies. I will tell you a little bit about them later. That the drug is able to do what it needs to do as a starting point. And it's only once we have these small studies with proof of clinical principle and proof of pharmacological principle with target and circuit engagement that we then move into the bigger clinical development phase, where we then, a lot more investment is required for large proof of clinical concept studies, and also then phase 3, which would hopefully then lead to approval and launch. So this is very high level what we do, and just to stress that biomarkers are essential at all stages of the drug discovery process. So you can see here some examples of translatable technologies that can be used and can be engaged both preclinically and also clinically. We can use, for example, EEG to demonstrate physiological responses. We can do that in a rodent test system. We can do that also in healthy volunteers. We can do that in patients. And this can give us an idea that the drug is able to do what we think it needs to do. But of course, there are also limitations with EEG in terms of where you can measure and so on. Very often with scalp electrodes and so on, it's not possible to measure in deep brain structures, and a different technology is needed for that, such as MRI. Now, PET is a wonderful technology, very challenging technology to identify new PET ligands, but it is the only technology we have that enables a measure of direct target engagement. It enables the elucidation of a quantitative relationship between drug exposure and binding to the drug target. And so this is a very, very valuable technology for us, if feasible, although very often, as I said, it's challenging to identify new PET ligands, but if that's available, it's wonderful. An alternative to that, in case direct target engagement is not possible, would be then something like functional imaging, where still we can demonstrate that the drug is able to exert a functional effect. And in this case, it's a more indirect measure, but nonetheless, we're able to demonstrate that the drug can work in the brain on the circuits that we've hypothesized to be relevant for a specific mode of action. And fMRI, this is functional imaging, very often combined with computerized behavioral testing. It's possible to combine imaging with measures of working memory, for example, where we can then introduce a cognitive load and really test how the circuit functioning differs with a cognitive load and then with the drug also on board. And impulsivity tasks would also be an example of that I will go on to discuss later, where these are also biomarkers, impulsivity behavioral tasks that can be used combined with imaging. And this is something that we will be using ourselves as part of one of our early clinical trials. Soluble biomarkers are also very, very important. Glycine is a good example of that, where you can take assessment from the CSF of biomarkers. These are very, very useful. Another example would be with a COMT inhibitor, which influences dopamine, and dopamine metabolites can be measured in CSF. And you can see a direct relationship then between dopamine metabolites, for example, with the degree of COMT inhibition. Tolcapone is an example there where this can be done. And you can then look at relationships between the soluble biomarkers in the CSF, also blood, of course, or other different matrices can be used. And it's possible then to demonstrate a change in biomarkers in CSF with then improvement in something like cognitive functioning, and then to correlate that then with drug exposure. So these are just some examples that there are tools available that can be used and are used to demonstrate that the drug gets into the brain and that it's able to exhibit target and or circuit engagement to really test and hopefully confirm our hypotheses. But I think it's an important but, that while these technologies are essential, there are still questions that remain, and innovation is still very, very much needed in order to increase the understanding as we move from these early clinical trials into later clinical development. And what we really want to do is to bridge between the DSM diagnosis, our increasingly robust understanding of circuit biology, and use then the biomarkers. And what is absolutely essential for these biomarkers is that they reflect circuit function, that they are scalable in clinical contexts beyond the very early clinical trials, which just engage tens of patients. We need to really identify biomarkers that can be used much later in clinical development in larger clinical trials, and potentially also, even as we move into a precision psychiatry world, also even in clinical practice. And very, very importantly, and this is something that is close to our hearts at Boehringer, that whatever we develop, that we are really taking a patient-centric approach to everything that we do, and that what we are measuring is things that matter to patients. So with that, I will move now into the example that I would like to use today, an area that we have been working in this area for quite some years now, and that is impulsivity. Impulsivity is an important across mental health. You see here in the upper panel here the definition of impulsivity. It is a predisposition towards rapid, unplanned reactions to internal or external stimuli without regard to the negative consequences of these reactions to the impulsive individual or indeed to others. On the left-hand side here, you can see that there are quite a number of DSM-5 diagnoses where impulsivity plays an important role. ADHD, of course, very common. Borderline personality disorder, also a great deal of borderline patients are highly impulsive. Bipolar, PTSD, and also addictions. So impulsivity is there, it's prominent and it's widespread. But what you see on the right-hand side here of some of the early patient engagement work that we have embarked upon, we've got to get some examples of quotes here from patients. And you can see from these quotes that even though we're focusing on one symptom where we believe we are reducing heterogeneity, what you see from these quotes is that there is a great deal of heterogeneity even when you focus on a single symptom. For example, an ADHD patient describing not being able to control their chaos in their mind, and that contrasting with a bipolar patient who describes drinking heavily and driving, knew that they were drunk but didn't care, wanted to do what they wanted. And then we have a borderline personality patient saying, they think that being able to make decisions more calmly would be helpful and would really change their life. And then finally, a PTSD patient describing attacking a family member physically and the consequences of that, living, staying with them forever, having to live with what they did and feeling ashamed. So even within a single symptom domain, a great deal of heterogeneity. And when you look then even within a single diagnosis, this is now using borderline personality disorder as an example. We know that borderline it represents a very complex behavioral syndrome, but again, it is highly heterogeneous. So you look at what behavioral or symptoms borderline patients often present with: affective instability, inappropriate anger, paranoid ideation or dissociation, and of course also impulsive behaviors. And impulsive behaviors for borderline patients can be reckless behavior, rage, impulsive aggression or violence, engaging in dangerous activities, things like sexual promiscuity, also repeated suicide attempts and self-harm. So it's a dominant feature of borderline, but also other psychiatric disorders, and has really significant impact on everyday functioning and quality of life. So, when you put it like that, it sounds like an almost impossible task. How could one even start to tackle the underlying neurobiology? Well, one of the reasons that we first embarked on impulsivity was because it has been studied for a very long time, and quite a lot is known about the underlying neurobiology of impulsivity. And research has identified that there are specific corticostriatal circuits that underlie maladaptive impulsivity. There are different types of impulsivity, waiting impulsivity, for example, or stopping impulsivity, or cognitive impulsivity. And using technologies such as functional imaging combined with impulsivity tasks, it has been demonstrated that while there are commonalities of corticostriatal impulsivity circuits, there are also differences that define then the different types of impulsivity. So armed with this sort of information, we are able to look very deeply then into these sorts of circuits and identify then potential drug targets that are within these circuits that we can then explore more carefully in our drug discovery processes. So just another word to the tools that we have for measuring impulsivity. There are circuit symptom readouts, both subjective assessments as well as objective assessments. And the subjective measures are clinical scales. Participants are asked to report on their own impulsive behavior, and using clinical scales such as the Barrett Impulsivity Scale or the UPPSP. So those are examples of subjective measures, but objective measures where patients are really able to demonstrate their impulsive behavior. And this then in what should be standardized, reproducible tasks. And examples of these impulsivity tasks include the 4-choice serial reaction time task, which assesses waiting impulsivity, or the stop signal task, which assesses stopping impulsivity. There's also the immediate memory task and the delayed discounting task, which also explore different aspects of impulsivity. And our goal, as said already, is that we are aiming to be able to use particularly objective measures that are scalable, patient-centric, and reflect impulsivity circuit function. And when we are then, when we have such tools at our disposal, we are able then to use these tools to select individuals for whom we think will have then the most likely chance of being effective. So we can use impulsivity tasks, for example, to select individuals that have been had the classical diagnosis of borderline or bipolar or PTSD or other impulsivity-related disorders. We can use these sorts of objective measures to identify those individuals with high impulsivity that we can then engage in our clinical trials. So that's the clinical perspective on it as a starting point. Very important for drug discovery is that all of this work can also take place in the preclinical setting. And for that, we are fortunate that many of these tools, clinical tools, have already been back translated. So the impulsivity tasks, such as the 4-choice task, or the stop signal task, they have these impulsivity tasks, clinical tasks have been back translated to the preclinical setting where rodents, for example, can be trained to perform impulsivity tasks. And the circuits that have been described to be relevant in the clinical setting have also been reproduced in the preclinical setting, where imaging and also other technologies such as oxygen sensors or glutamate sensors have been able to be placed into discrete brain regions of the brain, and circuits have been able to be explored also using technologies such as optogenetics to really then establish test settings where we are then able to screen for new drugs and explore then the efficacy of potential new drugs in improving behavioral outcomes in the preclinical setting, and also engaging the circuits that we have already shown to be relevant in the clinical setting, and to demonstrate that then preclinically. So this is how we then ensure the feasibility in the preclinical setting, which we can then, when we have a molecule in hand that we've proven to be safe and efficacious, we can then take it forward into our early clinical trials. But this strong translatability of these biomarkers between clinical and preclinical settings is absolutely essential. And just to illustrate some data that we have generated using these sorts of tools, you can see that here, where we have explored using an oxygen sensor the neurocircuitry that has been altered using one of our possible new drug candidates. And so we've been able to demonstrate that we're able to modulate a circuit of interest using this drug applied in the preclinical setting. And also on the right-hand side here, you can see an improvement in behavior. This is premature responses in the 5-choice serial reaction time task, where rats have been trained to perform on this task, and premature responses are considered to be the measure of impulsivity. And when the animals have been selected for high impulsivity, you can see the drug is able to reduce that impulsive responding. So when we then have used these sorts of methods to build up really a strong case that we are engaging the circuit that we believe to be relevant, that we are able to improve a behavioral outcome on a behavior that we believe to be relevant, we can then take that drug forward into the clinical setting. And what is very, very important as a first step is to ensure that whatever biomarkers we are using, that they are really properly established and as fully understood as is possible. And so for impulsivity, our approach was to first of all perform an experimental medicine research study as part of our stepwise approach. So what we wanted to do is to identify from the range of different impulsivity measures that are described across the clinical and preclinical literature, we wanted to establish a test battery in our own hands, because very often these tests are not standardized, protocols are not harmonized, And various publications use various different parameters for establishment of these tests. So the first step that we wanted to do was to perform an experimental medicine study to just establish this biomarker test battery. And we decided that we would take an ADHD patient population with a moderate severity, enriched for impaired impulse control. And we wanted to use then a drug that is already known to be effective in improving impulsivity in ADHD, and that is atomoxetine. And we wanted to test then the effect of atomoxetine on ADHD using a digital test battery, using the tasks that I've already described, and looking also at how the task performance and improvement in the task correlates with the clinical scales. And so that's a study that we have very recently conducted. What I'm sharing with you now is just some of the interim data that we've had. This is now on the left-hand side, you see the reduction in the number of premature responses in these ADHD patients and the 4-choice serial reaction time task, the waiting impulsivity. And on the right-hand side, you see the stopping impulsivity as assessed by the stop signal reaction time task. And what we wanted to do in this study was to look at whether or not the drug was efficacious following a single administration on day 1, and how that compared then when we repeated the study after 14 days, repeated the test after 14 days of administration. And you can see that the drug was efficacious both on day 1 and day 14. And this gives us then important information for how then to design the next stage of the clinical protocol, clinical development plan for our new therapeutic concept, where we want in the next stage to prove the clinical principle of a novel compound. This is then the phase 1b study. What we would like to do is then replicate to some degree what we've already done in our experimental medicine research, but also extend that. So we will be again including ADHD patients in this study, but also extending it to include also borderline patients. And these 2 patient groups will be used as independent cohorts. And the idea behind this study would be to test a novel compound that has gone through the preclinical tests as described, and to demonstrate then that in ADHD and BPD patients that we are able to engage the circuits that we've hypothesized to be relevant for impulsivity, and also to show an improvement on the behavior based on the objective impulsivity test battery that we've established. Another point to note about this type of study is that what we are aiming for here is to test the hypothesis that impulsivity behavior has a common underlying neurobiology that is independent of the DSM diagnosis. This we would be able to show with this novel compound, were it to be the case that it were effective in ADHD and BPD patients. And this is something that would give then confirmation at the earliest possible point in clinical development that one mechanism is able to treat impulsivity in ADHD and BPD patients, so 2 patient populations, and may also then give confidence that perhaps it could even extend beyond that into other highly impulsive populations, such as bipolar or alcohol use disorder, for example. So once we've gone then through this is then the translational medicine part of the studies that I am responsible for. We then move into once we've demonstrated in these early small trials. I mean, I should also mention that these sorts of trials are aimed to have about 40 patients. There will be 40, for example, 40 ADHD patients, 40 borderline patients, perhaps a crossover trial. It really is the sort of study that would just really demonstrate on a small scale that an effect of the compound and prove the hypothesis and give us confidence that the greater investment would really be worthwhile. And the greater investment comes with a proof of clinical concept study, where then we would work then with the individual diagnoses, borderline, for example, or bipolar, and endpoints in this type of trial would be primary endpoint could be an objective or a subjective impulsivity measure, and secondary endpoints could be something like measures of improved patient function, or perhaps also DSM disorder scales as required. So that gives you an overview of the type of approach that we are taking. Throughout this approach, it's very, very important to say, as I mentioned earlier, that patient insights are important for everything that we are doing, and we will be engaging with patients. It will be iterative as we engage with them throughout this entire process, as we engage with patients to ensure that whatever we are doing is really addressing the unmet medical need and is of clinical relevance. And I think that's also a very important point to make. The final piece of the puzzle is, of course, the relevance of these types of biomarkers, impulsivity lab tasks and scales, for real-world impulsive behaviours and ultimately functional impact. And this is something, of course, that we are very, very keen to show. We know that this is important, and this is some work that is now ongoing, where we are really looking now, working to really explore how we can best correlate an improvement with impulsivity on the laboratory or digital tasks with real-world behaviours such as those shown here, and then with really showing an improvement in things that really matter: employment difficulties, poor social functioning, low quality personal relationships, and so on. And that's, of course, a very, very important and difficult piece of the puzzle to fix, and this is something that we are also working on. So coming now towards the end of the presentation, we are moving away from the diagnostic criteria-led approach to enrichment. This is what I've presented to you today, this enrichment with circuit symptoms domain-led subpopulations, still very much in the DSM world, but really trying to enrich based on objective biomarkers. Important at this stage is the engagement with patients, as mentioned, but also with regulatory bodies, and I will speak to that in just one moment. And this is really important because we also at this stage to refine and validate the assessment measures that are going to be important for registration. And once we've gone beyond this stage, then there is the possibility to move then to the final stage of the precision psychiatry approach, which is really then symptom domain criteria-led transdiagnostic populations. But we are a little way off that yet, rather more in the middle box, with still work to do here before we can really move into something that would be more transdiagnostic in terms of the approach. So just a word then to the regulatory considerations. There are opportunities such as engagement with FDA, with the EMA, using meetings such as the Innovation Task Force meetings or Critical Path Innovation meetings. And these are opportunities to discuss novel approaches and concepts with the authorities. We have done that. We have discussed our precision psychiatry approach with both FDA and EMA, and we have discussed impulsivity specifically with them. And such meetings give the opportunity to have the discussion, give the opportunity for the authorities to provide feedback on potential hurdles, on concerns that they have, and also for them to give advice on the type of supporting data that would be needed. And the feedback that we have received so far from both of these authorities has been very positive. We have heard from both authorities definitely a recognition of the need to do things differently in order to improve the likelihood of success in psychiatry. We have also heard the willingness and openness to the use of biomarkers, of the use of impulsivity tasks, for example, as endpoints. But what we've also heard from them is that they would like us to follow the stepwise approach that we have proposed. They would like for us to bring to them the data that really shows that if you improve impulsive behavior on a task, that is, of course, ultimately connected with an improvement for the patient. And this is what we are now working on. So coming now just to a summary of using biomarkers towards precision psychiatry. What are then some considerations for this? Certainly, it's important to ensure a deep scientific understanding of the underlying neurobiology of the patient. This is something that we are really aiming to do. We are also aiming to use objective, scalable biomarkers that we can use to select patients and also demonstrate treatment response. As said, it's important that we can then establish the link between a biomarker readout and something that is really clinically meaningful for the patient. And very, very important is that because there are so many stages in this process, that there are really seamless transitions from research to translational medicine and then to later stage clinical development, so that we really are systematic and methodical in replicating and extending at each phase. And where possible, to explore and maximize opportunities for transdiagnostic development, especially where it is demonstrated that there is a common underlying neurobiology, then within a symptom domain, then there is also opportunity there for that. So now my final slide, I think also in terms of what's important for success, and that most certainly is collaboration. I think no one institution, no one company can really do this on their own. At Boehringer, we are really trying to collaborate as broadly as we can. We have quite a number of collaborations across the globe with like-minded individuals and institutions where we all share the common goal of trying to improve lives and trying to put the patients at the center of everything that we do as we try to improve the likelihood of success in the field of precision psychiatry. And with that, I would like to thank you for your attention, and I'm happy to take any questions or discussion points. Thank you.
Thank you very much, Janet, for a very inspiring talk. Happy to see new dimensions and new treatments coming along, so that's very good to know. I can't see any questions in the chat, so maybe some of you would like to raise their hands and open their microphone. Bruno?
Many thanks for your very nice, clear, and didactic intervention talk. I have just a question. Could impulsivity also be considered as an phenotypic marker of the disease that therefore remains stable over time, regardless of the treatment strategy proposed for managing the disease?
Right. It's possible. I think it depends, and it has, of course, we don't know. But one could imagine that were one to improve impulsivity, that with time, if somebody is better able to control their impulses, that it may have long-term consequences on other aspects of the disorder. It remains to be proven, of course, but one could imagine that that could be the case. So if you imagine that somebody is better able to control their emotions, their anger, for example, that maybe they are better able to have meaningful relationships, they are less likely to give up their job, and there may be then a positive cascade that ensues as a consequence of that. So yes, one would hope so. Yes.
Great, thanks.
So maybe while you all think about questions, I have two questions, in fact. First, I wondered why you don't have any blood-based measures for impulsivity screening.
Right. So it's possible we could, and we would take blood actually, but what would be important would be that we were able to demonstrate the correlation so that we have a good sense of what it means if you change something in the blood, what that means in terms of relationship to what you're changing in the brain. But it could absolutely be blood. It could be a blood-based biomarker, and that would, of course, be the simplest thing to have. So yes,
because to my knowledge, one of the first biomarker ever described was 5-HT.
yes.
So maybe 5-HIAA, for example, could be a very simple tool. So maybe difficult to use because you have to control for what patients are eating in it. But, be very simple to use and maybe less expensive than the ones that you have described for future clinical use.
Yeah, fully agreed. Absolutely. It would just be that link that would need to be established, but it could be established, and then it would make for a very, very useful, inexpensive, scalable biomarker. Absolutely. Agreed.
And following up on the cost associated to all these measures, so we start hearing, we meaning all of us working on the field of precision psychiatry, criticisms, as usual, about possible expenses and costs associated to all these tools that we are developing to improve precision of psychiatric diagnosis. So what is your vision about the availability, the capacity of different countries where resources are quite scarce to use these markers?
Yes, it's a very, very important point, of course. It so of course cost is essential. There's no question about it, and cost has to be considered. And therefore, we are looking to establish surrogate biomarkers that would be then scalable and not so expensive. And if you think about digital technologies, I know it's not accessible to everybody, but where it's accessible, these can be inexpensive. Expensive because it could be that remote monitoring is possible. So it's not necessary for somebody actually to have a face-to-face meeting with a psychiatrist or a physician. It could be that some measures are possible at home, and that could reduce some costs. So, of course, it's something that has to be borne in mind, that the costs cannot be overlooked, and we need to ensure that there would be access to all. Absolutely.
And in your vision, do you think that research will be needed to describe and define some clinical biological signatures, but then at the end, we might end up by something which is maybe user-friendly and less expensive than what we use in research?
That is absolutely the vision. Yes, exactly. Yeah,
okay.
thank you for those questions.
For hours for questions. So anybody willing to, hope I didn't miss any hands up or... Okay, then maybe one more question while we all think about other questions so you have had one slide about regulators, and of course, I wanted to raise the question, but you answered it. But same question for professionals, for example, or even patients. How do you plan to change their vision of what is depression, what is schizophrenia, and why we need to target dimensions? How do you want to change their representation?
Right. So we have to engage with them. I think the same is true also for payers. We have to engage with all of these stakeholders all the way through. Now, our impression so far is that patients, and maybe somebody in the audience here would be I'd be interested to hear if this is also your experience. What we've heard so far is that patients are they like to hear that there is an explanation for what they are struggling with. They like to hear that the problem that they are struggling with is associated with something that is going wrong in their brain that can be described. So that they have a sense that it will remove stigma. If it's not just how they are, but it's because something is not right in their brains, and that this is something that can be treated, and that they can start to think of a brain disorder like any other type of disorder. It's something that is going wrong and that can be treated. And so, so far, the feedback we've had is that that patients would welcome this, and that it won't be too difficult to convince patients that that this is the case, that they would be open to hearing about the biology. And we know now from the use of smartphones, for example, that people love data. People love things that they can measure, even if they're on their smartphone, they just have their health data, how many steps they took in a day, or how their sleep was, or whatever. People like data about themselves. And if they could have data also about something that's going on in their brains that maybe explains something that they are struggling with in their lives, then our expectation is that this would be something that they would welcome.
Okay, very clear. Thank you very much for these answers. Any remaining questions to our host today? Oh, no.
I've just a short question. For you, impulsivity in bipolar patient are marker of the disease or addictive comorbidities or suicidal risk?
Oh, gosh. There is certainly suicidal risk there, right? Yeah. Absolutely. This is a concern for sure.
Or addiction, also, of course.
Absolutely. Yeah, absolutely. I mean, it's all in there together, I think. It's difficult to separate those things out. And then yeah, that's a difficult one, I think.
Great. Thanks.
Okay, well, with that last question and answer, we just have now to thank very much Janet for a wonderful discussion and conference. Thank you all to have attended to this meeting, and we give you, I can't remember when is the next conference, but I'm sure you have that on your flyer describing the different conferences. So see you soon and have a good day.